Hello to everyone. First of all, I want to thank the organizing team of the conference for the opportunity given to take part once again at this event. My name is XHULAJ. I am a researcher at the Institute of Plant Genetic Resources, Agricultural University of Tirana. On behalf of my colleague, we want to represent you the research title Analysis of Genetic Variation in Bred Wheat by Grain Yield Components. Our country is an area rich in crop biodiversity, with 24% of total area in agricultural land. Agriculture is still a significant sector of the economy, which contributes to 22% of the GDP, providing the income base for most of the population, with approximately 58% of it employed in the sector. Despite economic hardships in Albania, wheat yields have continued to grow at an average annual rate of 1.6% since the early 90s, and nowadays, bred wheat is the most important cereal crop and plays a vital role in food and nutrition. Our institute, Institute of Plant Genetic Resources, former Albanian Gene Bank, contains more than 594 accessions of wheat germplasm. The wheat collection is represented by wheat forms as Triticum aestivum, Triticum durum, Triticum turgidum, and its wild relatives. And this institute, as a gene bank, offers the main means to store plant genetic resources, to protect genetics materials, and also to provide the raw material for the plant breeders. The aim of the study was to provide a clear assessment of the variability and to evaluate possible interrelationships among yield contributing traits through characterization of 18 accessions of wheat germplasm, to identify genetically divergent wheat genotypes with desirable traits that can be used as donors in breeding programs. The plant material used, object of the study, are part of the base collection. The genotypes numbered from 1 to 18 have their accession code AGB for Albanian Gene Bank, accession names, origin, and the date acquired in our institute. Some of them are more than 20 years in conservation minus 20 degrees. The plant material is characterized by testing on the field and in the laboratory. The field experiment was conducted during the autumn season 2017-2019 at the experimental station of Institute of Plant Genetic Resources, Agricultural University of Tirana. Morphological characterization of the accessions was conducted according to international standards for gene banks of Institute of Plant Genetic Resources, the wheat descriptors used. Despite genetic composition, also climate factors as rainfall and temperature was taken in consideration during the two growing seasons, as these two factors plays an important role in determining crop growth and influencing also grain yield. All the data were statistically calculated by analysis of variances. Distances between genotypes were calculated by clustering method, principal component analysis, and also correlation coefficients between characters were taken in consideration for all the 9 quantitative traits. Our results suggest that significant variation was found in quantitative traits as grain yield, number of seeds per spike, weight of seeds per spike, and weight of 1,000 seeds. So, wheat genotypes used in the study reacted differently in the two growing seasons, giving high results in most of the traits that contribute directly in grain yield during the first crop year. During the second year, the wheat genotypes presented high values only for plant height trait. We observed that all the wheat genotypes produced higher grain yield during the first season in compared to the second growing season. In terms of total precipitation during the entire growing seasons, there exist differences between the 2 years of crop growth, where main rainfall is observed during the first year and a lower value of precipitation during the second crop year. So we observed that the highest grain yield during the first year in compared to the second one was associated with the highest vegetative precipitation and air temperature also, and with highest reproductive stage precipitation. ANOVA analysis found the presence of significant differences between wheat genotypes for most of the biomorphological characters. The results show that genotypes with the highest grain yield, as genotype numbered 4, 5, 7, and 11, were those with high values in traits as tiller capacity, weight of seeds per spike, weight of 1,000 seeds, and number of seeds per spikelet. According to this analysis, strong positive correlation is observed between grain yield and number of seeds per spikelet, tiller capacity also, weight of seeds per spike, and weight of 1,000 seeds. Correlation analysis is useful to identify agronomic traits that are positively related with yield. According to the multivariate correlation analysis, different traits exhibited positive relations between them, as tiller capacity of the wheat genotypes with weight of seeds per spike and weight of 1,000 seeds. Strong positive correlation is observed between weight of 1,000 seeds and weight of seeds per spike, and also between the number of seeds per spike and weight of seeds per spike. We observe strong negative correlations and among traits as number of spikelet per spike and most of other traits as plant height, tiller capacity, weight of seeds per spike, and grain yield. And revealed that the 18 wheat genotypes were grouped in 3 major clusters. The first cluster included 10 bred wheat accessions, similar for different biomorphological traits. The 2 wheat genotypes within this cluster with the lowest level of similarity observed were genotype 1 and 3, or named Generoso and Nestos. The second cluster grouped 5 accessions where genotype 13 and 17, named Universe 2 and Universe 6, presented the lowest distance among them for similarity in tiller capacity and number of spikelets per spike. And the third cluster included only 3 wheat genotypes. Subclusters at this group were with the lowest level of dissimilarity observed not only within this cluster, but also among the 18 wheat genotypes. Part of this research are genotype 7, or Dodonic cultivar, and genotype 11, or ICPA 05 cultivar. So these 2 accessions presented similarity in traits related to grain yield as weight of 1,000 seeds, number of seeds per spikelets, tiller capacity, and spike length. The principal component analysis suggests that 75.51% of the variability is exhibited among 18 common genotypes, wheat genotypes, by 3 components, where the 2 first components influenced mostly the variability. The first principal component at the level of 48.82%, and the second one at the level of 15%. In addition to cluster biplot suggests that and explain the percentage variance associated with each principal component, and the vector view of the biplot suggests a strong positive correlation among traits, as indicated by the small obtuse angles between their vectors, and especially between weight of 1,000 seeds of the genotypes and weight of seeds per spike, where the correlation between spike length and other traits as plant height, weight of 1,000 seeds, and finally grain yield was near 0, as indicated by the near perpendicular vector. According to the principal component analysis, The variability presence within the first component was basically related with traits as weight of seeds per spike and weight of 1,000 seeds, also by plant height and ground yields, but this principal component was poor in variability for number of spikelets per spike. And this last trait, followed by spike length, strongly contributes at the genotype's variability in the second component, but this PC was very poor in number of seeds per spikelet and in tiller capacity. Where in the third principal component, the positive effects exhibited trait as number of seeds per spike and number of spikelet per spike. According to this analysis, the variability that affects most within this component, within the three components, is number of seeds per spike. In conclusion, the results of this study succeed in obtaining an important scientific information on which plant genetic resources that are in storage in our gene bank. The study identifies traits with agronomic interest that account for genetic diversity and which will facilitate the maintenance and agronomic evaluation of the wheat collections. The traits with more significant weighting on respective PC variants can be utilized successfully as quantitative markers for further evaluation and characterization of the wheat germplasm that is in storage in our gene bank. Similarity among some of wheat genotypes found in this research in each cluster could be explained by common parent origin in their pedigree. Accession analyzed can be selected and utilized for sustainable wheat breeding programs. Thank you very much for your attention.